Welcome once again to another MOOCs lecture of BP 103T, that is Introduction to Semi-Solid Dosage Form. We are covering Unit 5 and we are going to understand the semi-solid dosage form in a bit detailed manner. I am Anjali Mishra, Assistant Professor at Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. So, let us start. So, before understanding the mechanism of dermal penetration of the drug, let us understand that what we are going to understand here. We have actually started learning about semi-solid dosage form. So, we have actually started learning about semi-solid dosage form. So, semi-solid dosage form, as I have said in the previous slides, semi-solid dosage forms are actually applied topically onto the skin surface or onto the mucosal surface. So, it is important to understand for us that these semi-solid dosage form are actually going to penetrate the skin surfaces onto where it is applied, because eventually what matters to us is the penetration of the drug from the surface where it is applied, because as the drug penetrates into the surfaces, the action is evoked, and whatever action we want, for example, if we wanted antifungal action, we get that antifungal action. If we want emollient effect, so if we get that emollient effect. So, effect is very important in order, so for that purpose the penetration of the drug is also very important. So, here in this entire presentation we are going to understand that how and what are the various routes through which dermal penetration of the drug occurs. So, here first of all we have a diagram of skin. We are supposed to understand that how through these different parameters our drug actually penetrates the various parts here. So, before understanding anything, let us understand this diagram. The skin diagram, this is nothing but the topical surface onto which is exposed outside. This surface is exposed outside. In this surface we have got small openings through which we have got our hairy structure. This is nothing but the shaft or the base of the hair structure. We have so many pores here through which we get our sweat and sebum and all sort of skin secretions are produced, and this is the upper surface. Now, moving ahead, we have got three important layers. The very first initial layer is nothing but the epidermis. Epidermis consists of granular cell. It consists of stratum corneum. Another thing, it consists of spinous cell layer and basal cell layer, and another thing that it consists of sebaceous gland. Sebaceous gland is responsible for the production and oozing out of sebum that usually keeps our skin hydrated. So, this is all about the layer of epidermis. Now, below epidermis there is another layer known as the dermis. Dermis consists of erector pili muscle, sweat glands, nerve endings and hair follicle. So, majorly the hair follicle, the nerve endings, the pili muscles are present in the dermis layer of the skin. After the dermis layer, the next layer that we have is the SC layer, oftenly called as the SC layer or the subcutaneous layer, that consists of collagen or the elastin fibers or artery, veins and fats or the adipose tissue that usually gets converted when we have excessive production of glucose there and glycogen. So, that gets converted into fatty tissues, and that usually gets accumulated beneath the surface of the skin and specifically onto the which surface, onto the SC layer, subcutaneous tissue layer. Hence, this was all about the skin and different layers of skin. Whenever we are applying anything onto the external surface of the skin, that is onto the stratum corneum of the skin, the dermal penetration occurs and it eventually reaches this layer, that is the artery, vein or to the SC layer. Actual exchange of the medicaments and the blood takes place here between the sweat gland, between the nerve fibers or into the artery and vein, wherever it is supposed to actually exchange the substances. So, it is important that when we are applying anything onto the external surface, it passes these layers and reaches the main target organ or the main target area from where actual exchange of medicaments should take place. So, this is all about your skin's diagram. So, we know dermal penetration means what? It is going to insert or it is going to penetrate through the layers of skin only. Another thing that the skin is very effective in selective absorption of drug. Yes, the skin is very effective in selective absorption of drug. Whatever substances are to be absorbed, that is actually being selected by the skin. If the skin feels that yes, I need to absorb this medicament, then only that the skin absorbs it. If it does not find it appropriate or fit, it oftenly gets rejected there. So, skin plays a very important and selective factor in absorbing the different types of drugs. Then epidermis layer plays a vital role in controlling elements for the drug penetration or the dermal penetration. Just now I have said that the skin know what to select and what not to select. So, actually which part of the skin plays an important role in selecting the ingredient? That part is nothing but your epidermis layer. As I have discussed, the skin consists of 3 layers: epidermis, dermis and subcutaneous layer. So, epidermis layer is the major important controlling layer that actually controls at what amount and how much amount or what kind of medicaments must pass or enter the actual systemic circulation on application of any medicament onto the stratum corneum. Now, studying of dermal penetration of drug is important because when we are applying or when we are formulating semi-solid dosage form, it is oftenly going to show its action topically onto the skin surface or onto the mucosal surface. So, it is important that to understand that how does this penetration occurs, what actually happens. We are going to understand these sort of mechanism into the next slides. So, mechanism of dermal penetration occurs majorly by 3 routes. The very first route is the transfollicular route. So, it is a transfollicular route. Another type of mechanism of dermal penetration may occur from transcellular route. So, it is a transcellular route. Then the last mechanism of dermal penetration may occur from intercellular route. Intercellular route. So, we are going to understand these 3 different types of dermal penetration in a detailed manner. So, let us understand the very first, that is the transfollicular route. Follicles are nothing but they are the minor openings or the follicles of hair, follicles of sebum through which the dermal penetration of the drug occurs. So, this is the shortest pathway for drug passage and provides larger area for drug diffusion. So, if we want to produce a fast and a quick action, the thing that is going to give a quick and rapid action is nothing but the transfollicular pathway. So, transfollicular pathway gives a very quick rapid action, it is the shortest pathway, and at the same time it has a very larger area for diffusion, because until and unless diffusion occurs, we are not going to get the biological response. So, that is all about your transfollicular pathway. Another pathway is the transcellular pathway. So, the name itself is very much self-explanatory, that is transcellular. So, between the cells. So, drug here passes through corneocytes, which are hydrated hydrophilic pathway. So, here majorly hydrophilic drugs is going to pass through these, because the pathway here is hydrophilic in nature, and the drug passes through between the cells. For example, if I have a corneocytes here, there are multiple corneocytes present onto the layer of the skin. So, between these 2 corneocytes there is a layer through which our medicament or the dermal penetration might occur here. So, that is nothing but your transcellular route. Another mechanism through which dermal penetration may occur is the the intercellular route. Intercellular route. Now, here I was talking about the between 2 cells. So, here the penetration occurs, which is nothing but the corneocytes. But in the intercellular route, the penetration or the passage of the drug occurs inside the cells only. Here, this is very important to understand the difference between transcellular and intercellular. Here, drug diffuses through continuous lipid matrix that is present between the cells. So these are some of the pathways through which the dermal penetration occurs. First is your transfollicular, the another one is transcellular, and the last one is transintercellular. So transfollicular means it is the shortest pathway through which easy and fast diffusion of drug occurred, and at the same time the diffusion produces a larger area. The diffusion matrix gets a larger area for exchange of medicaments or for the exchange of nutrients or the biological responses. It gets a huge area for that purpose. Now next type is nothing but your transcellular route. Transcellular routes occurs between two corneocytes. Corneocytes, between two corneocytes there is a place, and that place is nothing but hydrophilic pathway in nature. So any drugs that are majorly of hydrophilic origin or aqueous origin may find it very easy to pass through these type of pathway. Now another pathway is the intercellular pathway. Intercellular pathway is majorly favored for lipophilic drugs. Here it is majorly favored for lipophilic drugs. Any drug which is of organic in nature, which has a fatty origin in nature, which is a lipid soluble drugs, may find or may prefer the intercellular pathway, because the pathway is majorly made up of lipid matrix. So lipid matrix make it easy or make it feasible for the mechanism of dermal penetration of any of the lipophilic sort of drugs. So this is all about the three different types of mechanism of dermal penetration. Now here is a diagram which represents that how and which route actually passes from which area. So when I say that this is a layer, that this is nothing but it is a upper layer of the skin, that is nothing but the epidermis layer of the skin through which the transportation or through which the penetration of the drug occurs. Here we can see the very first thing is the transcellular route. Just now I have discussed transcellular route means what? Between the cells there is a production of corneocytes, and it is a hydrophilic pathway. So from where the for example if the API is present here, so API is going to pass through this pathway and it is going to actually reach the dermis layer or to the below layer, that is a subcutaneous layer. So this is nothing but the transcellular route. Then another one is the appendageous route. It is oftenly called as transfollicular route. It can also be called as follicular route. Now follicular route is actually as a result of follicles that are present. That follicles can be your hair follicle, it can be your the small sweat glands that are present onto the surface of the skin, through which actually the transportation of semi-solid dosage form might occur. Now as you can see, this is a hair shaft. So this is the base of the hair and this is the shaft of the hair through which there are certain openings and there is certain space through here. This space is actually occupied by anything that is being applied onto the skin surface, and this pathway is nothing but the transfollicular. Here you are saying the appendage route, which is also known as transfollicular route, and this is the fastest pathway through which we can actually cause the dermal penetration of the drug, and fastest pathway, but at the same time the area for diffusion is very much high as compared to your transcellular or through the intercellular pathway. In the same manner, we can see that there is an intercellular or extracellular matrix route. Just now I have discussed in the previous slide that intercellular or the extracellular pathway is a lipid profile pathway. It actually helps lipid drugs, lipophilic drugs to pass easily through that. So here we can say that due to the difference in the charges of the skin surface and to the application surface or to the applicant or to the substances that is being applied, the ions may get diffused. The ions may get diffused. This is nothing but it is the lipid profile pathway. So it is entirely lipid profile pathway. So can I say that this transcellular is aqueous profile pathway? So if we are formulating a drug, if we are formulating a medicament that is majorly actually aqueous soluble in nature, so which pathway that is going to prefer? Definitely it is going to prefer the transcellular route. If the formulations that we have made is majorly of the lipid in nature, it is going to go for intercellular route. However, if you want a quick diffusion and the proportion of oil and water is same or it is equal, then we can definitely go for ependygeal route, or it is also known as transfollicular route, and the mechanism or the penetration of or the dermal penetration is going to be very quick, and at the same time it is going to get a very high and larger area for diffusion. So, understanding mechanism of dermal penetration in a more deeper way. First of all, low molecular weight molecules penetrate very easily when at the same time when we go for high penetration molecule. If we want to actually penetrate or to actually transport a medicament which is of low molecular weight, it is very easy for us because low molecular weight compounds are easily transported because the size is less, and at the same time it can be easily facilitated or it can be easily transported by any of the route that we have just now discussed. Now, skin appendages are main route for electrolyte, for polar steroids, for antibiotics and for colloidal particles. So, skin appendages, that is the transfollicular route, may help in the transportation of any of the charged ions, like it can be electrolytes, it can be polar steroids, it can be antibiotics, and it can be colloidal particles as well. So, if we are making any formulation that has the presence of electrolytes in it, we are very much sure that which pathway we are supposed to accept, or which is a major pathway for dermal penetration. That is nothing but the skin appendages pathway. Then, hair follicle route is important for penetration through the stratum corneum. We just now saw that if this is the hair follicle route and this is the region and this is the shaft, it is easy for us to make, or it is easily for us to penetrate the drug through the openings that is present here. Then topical agents may form a depot or reservoir by attaching them with stratum corneum for longer duration. Now, topical agents may also form a depot. Depot is nothing but it is a reservoir of medicament that is present here. So, if we want that the patient is actually experiencing a pain for a longer period of time and is not in a state to apply that medicament himself by herself again and again. So, what we are going to do here is we are going to prepare a semi-solid dosage form in such a way that actually has a depot or reservoir. So, whenever if there is a skin surface and on that if there is a depot or a reservoir, this depot is going to release, or it is going to ooze out the medicaments slowly and slowly, and the medicaments released is going to produce action for a longer period of time. So, what here happens, the residence time of the drug increases, the efficacy of the drug increases, and at the same time the compliance of the patient also increases. Then average residence time of drug in dermis may be because of period of 1 minute before it gets washed away. So, the average time taken by a topical agent to produce or to get engulfed in it on the dermis layer is actually 1 minute. After 1 minute it gets immediately washed away, or it enter into the next layer, that is the SC layer or the subcutaneous layer. Then next is NSAIDs, that is non-steroidal anti-inflammatory drugs, reach far down muscles to form depot. For example, if we are applying a painkiller, any ointment onto the skin surface, it is actually if it belongs to NSAIDs, what happens here is it enters quickly into the SC layer and it forms a depot there. So, what happens by the formulation of depot, the medicaments is released slowly and we get a constant or sustained release action here, which is a very important factor when it comes to topical agents. Now, when we are dealing with fundamentals of skin permeation, it is important for us to understand the equation which is represented here is rate of permeation. Now, rate of permeation, rate is nothing but when anything happens in a particular time. It is oftenly represented as dq by dt is equal to Ps Cd minus Cr. Okay. So, here the medicaments or the equation that here is representing is dq by dt is Ps then it is CD minus CR. dQ by dt is nothing but it is the rate at which the dermal penetration of the drug occurs, and here PS is nothing but it is a permeability coefficient, and here CD is concentration in the donor compartment and CR is the concentration in the receptor compartment. So, let us take an example. If there is a compartment here and there is a semi-permeable membrane, this membrane can be of anything, it can be of nitrocellulose, it can be of animal skin, previously it is used to have animal intestine. Nowadays there are cellophane membrane, nitrocellulose membrane, and let us consider that this is a donor compartment and here this is a receptor compartment. So, from the donor compartment our medicament is going to enter into the receptor compartment. So, at what rate the permeation is going to occur, it is definitely being decided by the rate, okay, and this rate is actually represented by an equation dQ by dt is equal to PS CD minus CR. If the rate is high, it means the dermal penetration occurs at a quick rate or at a quick pace. Now, as we all studied about the dermal penetration, how it happens, what are the factors that is affecting dermal penetration, we should also understand that what are the major factors that actually governs the penetration of dermal preparations or the topical preparations. Majorly it is classified into two groups, biological factors and physiochemical factors. So, we are going to study each factors in a detailed manner. So, let us understand about biological factors. So, what are the biological factors that actually differs from an individual to individual and is actually responsible for the increase in dermal penetration or for the decrease in dermal penetration? Let us see. First is skin condition. Definitely, if a person is having a skin which is dehydrated, so for that purpose the dermal penetration is going to get affected as compared to a person whose skin is hydrated. So, depending on varying the skin condition, the factors may change. Then skin age, definitely if the skin is of an older patient, then the factors is definitely going to be affected as compared to a patient that is a young patient. Then blood flow if to a particular skin is higher, then quick exchange or quick penetration may occur as compared to a skin which has decreased blood flow. Then we have come across a regional skin site. Regional skin site means a site where actually it differs from area to area, but in a particular area of our body the blood flow is going to be high. So, for that purposes the availability or the permeation for that region is going to differ as compared to another region which has got a less blood flow. Then skin metabolism, if the skin is obviously exposed to a higher metabolism, then the penetration might change. If the skin is exposed to a lower metabolism, the factor or the engulfing factor is going to differ. Lastly, species difference. Yes, if the we have got various species, if we talk about humans as well and animals as well. So, depending upon the species, the penetration may vary, the action may vary, the bioavailability may vary. So, these are some of the condition or these are some of the biological factors that definitely influences the rate of permeation of drug. Here we have another factors which we are supposed to keep in mind while formulating a topical agent, that is nothing but physiochemical factors. Now, physiochemical factors plays a very vital role if we want to formulate a topical dosage form, because by understanding these factors we can actually change the medicaments or we can change the factors that influence the medicament. Under that, first thing is skin hydration. If the skin is hydrated, definitely the action that is going to produced is quick and bioavailability will be high. Another is temperature and pH. So, temperature and pH plays an important role in deciding the bioavailability and the penetration of the particular semi-solid dosage form. Then diffusion coefficient. If the diffusion coefficient is high, the permeation or the penetration factor is going to be high. If the diffusion partition or the partition coefficient is low, definitely we are not going to get a high diffusion. Then drug concentration. If the drug concentration is high, it means that the drug or the formulation has penetrated a particular pathway. If the drug concentration is low, it means that the medicament has been exposed to certain conditions where the drug was degraded. Another physiochemical factor is a partition coefficient. Partition coefficient is the solubility of a particular drug in a aqueous or into an oily solvent. So, depending upon the type of medicament that we are formulating, the partition coefficient is going to vary. If it is an oily soluble drug, then the partition coefficient must be selected, or the vehicle must be selected as per the requirement. Then the last physiochemical factor is the molecular size and shape. This plays a very vital role in deciding the permeation of drug from the stratum corneum to the subcutaneous layer, because if the size is too high, then the molecule is not going to pass through the passages and there is going to be difficulty. So, molecule shape and size plays an important role. So, that was all about the dermal penetration of the drug through various layers. I hope this session was very informative for all of you. Thank you so much.